Okay, so today, let me activate everything that we're going to be doing today. First thing is extra credit. If you can get someone to take pictures of you doing the experiments right now, I'll give you extra credit. So if someone takes a picture or you take yourself a little selfie, that and you upload, then I will give you extra credit for that. You can even take a picture of like your setup of everything. You can see I have my setup right now here. I'm going to be doing the lab and you can see me whatnot a little bit later. So if someone in your household or yourself can take selfies of you conducting the experiment, that would be great. Okay. Next thing is the stormwater lesson itself.
So, Millie, I have a question.
Yeah.
Do we get extra credit for yesterday?
I don't know. I'm thinking about it. Okay, so this is just kind of what we're going to be doing today. Then in your modules, you have 3 things activated. One is like the overview of today's lesson, the doc. The other one is what you got to fill out while you're doing your experiments. So watch it load. So the first link is just kind of like the Google Doc. Oh, yeah. The second one is a step-by-step instructions. Okay, you might want to have that open on another screen. Then the data sheet. So let me show you the data sheet right now. This is the data sheet. It looks like your other one, right? But this is your testing one. You're going to be testing 1, 2, 3, 4, 5, 6 times. 1, 2, 3, 4, 5, 6. Yeah, 6 times. We're going to do 6 different experiments. 2 of them require time. One of them's 48 hours, the other one's 5 days. The other ones you can do right now. And you're going to check and highlight. If you can't check, then just highlight, right? Using the highlighting tool. You're going to tell me the numbers. And I'll let you know how to use the scale in a second. Then I want you to answer at the end, what results surprised you the most? Why? Answer must be in complete sentences. Okay, page 2. So you're gonna need to take some pictures. This is not extra credit pictures. These are point pictures where you're just gonna take pictures of your results. So this one, you need 48 hours. So this one, yesterday, the kids who went into the lab yesterday, we did this one and I brought it home because I need to check the results tomorrow. But this is what we have so far, under 24 hours. So I can't take a picture of this yet because it hasn't been 48 hours, right? So 48 hours. So when you have completed the dissolved oxygen lab, you're going to take a picture, then you're going to upload the picture right in here. So you're going to insert the picture here. I'll show you how to do that a little later, okay? So you might want to have this. So right here, it's like water appearance. So right now, as I hold my water bottle, does it appear clear, slightly cloudy, opaque? So you're going to get your own copy of this sheet. You're going to highlight, however you want to fill it out. And then when you're done with it, which probably won't be until, it won't be until 5 days from now. I might change it. I might have you turn it in once with the first answers from today, and then turning it in again once you do the 5-day experiment, which will be Tuesday. All right. Okay. So then the last link that we're going to open is the step-by-step instructions. So the step-by-step instructions is, it's like a Google slide. So super important, do one test at a time, read each step one at a time. So read step 1, do step 1. Read step 2, do step 2. Read step 3, do step 3, okay? Don't try to read all of them and then do it. Like, you got to read one step at a time. Something I learned yesterday is like, I didn't do that and I almost messed up one of the experiments. But one of you read and you saved us because you did it correctly. And I was thinking we were supposed to do something else. So props to you, Rochelle, for reading, because I did. Okay, are we ready? So have whatever tabs you need to have open. because we're ready to go. Okay, so first one we're gonna do is we're going to do the BOD, biochemical oxygen demand. This one will take 5 days, which is why I want to start it first. In slide 3, you are going to see on the directions. Now, you have access to this. So my suggestion would be to open it if you want to. I would. And then figure out, let's see if I can do this. Okay. I don't know if it's really hard to see on your screen. So first things first, fill a small test tube to overflowing with sample water. So how do I, let's see if I can't. Okay, so I'm just trying to figure out like how to do this where you see me and everything. So first one's first. Okay, find your BOD. That's the one with the aluminum foil in it. It's the top left. It says BOD on it. BOD. I don't know what. It is of most importance to have this Google slide open. You're gonna have to read the directions. So I only see one of you on it. Oh, two of you. Okay. Because I think it's just better for you to see it. Here we go. Okay. So you're gonna fill the test tube. Here's how you fill it. Totally wear your gloves. Take your syringe. Take your syringe, open the syringe. I have a plastic bag to protect my counter because it's dirty water. You can also, if you don't have this, you can just make sure you clean up after yourselves, okay? All right, so I'm gonna take the syringe, okay? Now, gotta show you how to use a syringe. You open it up, have it like all pushed out, right? You're gonna take this, you're gonna submerge it into the water, and then once it's inside the water, then you pull up, and that will fill the little tube with water, okay? So you take the syringe, and you might need to like hold it at an angle and fill it up with water by pushing up. So now my syringe is full of water. You're gonna take your BOD, and I'm doing step 1, overflow. So I'm just gonna kind of put it over. I'm gonna put it over this, and I'm gonna fill it with the water. And it wasn't enough water, so I'm gonna fill it up more with water. Submerge it, pull it up, pull the syringe up, just like so. Fill it up with water all the way to the top, it says. Okay. Fill a small tube to overflowing with sample water. So then it says cap, so I'm gonna put the cap back on. Wrap the tube with aluminum foil. So I didn't put any tablets in here. This is why I want to do this one first. All you do for the BOD is just fill it with water, put the cap on, take the small piece of aluminum foil that came in your kit, open it up, okay, and you're gonna wrap it. There it is. So I wrapped it. I did not put any tablets in here. There's no pills in here, no chemicals in here. Now, this is going to be like this for 5 days. Friday, Saturday, Sunday, Monday, Tuesday. On Tuesday, when we have class together, we're going to unwrap this and we're going to add the 2 tablets that are in your kit for BOD. So right now, this is it. All you do is I'm just going to put it somewhat in my, like, I'm just going to stand it up in my little kit, just like that. See how it's just standing there? Just that, I'm going to do that. So the whole point of wrapping is to get rid of all the sunlight. So there's no sunlight hitting it, not even artificial light. We're done. Okay, so that's the first one. We're okay? Give you guys like a minute to catch up. All I did, take BOD, fill it up, cap it, wrap it. That's it. Okay? All right. We're going to move on to the second experiment. Okay, so the second one takes 48 hours. That's this one that I have right here from yesterday. I cannot tell the results until 24 hours from now. So you guys will be Sunday. So on Sunday, you're going to see what this looks like on Sunday for yours. Okay, so let me go into the directions for the chloroform. Right. Okay, let me zoom it in. So we're on the second experiment. Fill the tube to the 10 millimeter line, okay? So the big plastic tube already has the tablet inside and it already has the mark of 10 millimeters. Yay, so it's gonna be super easy. So first step, fill the tube to 10, okay? So we're gonna uncap it. Take our syringe, grab some water. Fill it to the 10. Fill it to the 10. Replace the cap, step number 2. Stand the tube upright. So make sure with the tablet flat on the bottom. Okay, so we're just gonna stand it up upright like this. It doesn't even shake to shake. Reading the directions. Incubate the tube upright at room temperature for 48 hours. Store out of direct sunlight. Okay, so you're just gonna store it anywhere around your house that's not near a window. Okay, just store it like this. You can even put it back into the back into your tray, but standing up. All right, so you can just store it like this. I have the other one too. Just be careful. Right there. That one's 48 hours. Okay, so this one's Sunday. I don't know if you want to put a calendar reminder to look at it on Sunday. So this one, you're going to check to see what color it is on Sunday. Okay. And then on Sunday, then we're going to see our results, if it's yellow or red. The one we did yesterday, it seems like it's going to be red, which means liquid above gels. Oh, look, there's some gel. So this will tell us like the results, if it's positive or negative. Many gas bubbles present. So this one tests for poop. like dog poop, bird poop, poop. So this one's poop. This one's you're testing for poop. Yeah, your water can have poop. That's why you were wearing gloves. It's not pretty, but it's science. All right, are we ready? Or do I need to give you like a couple of minutes to catch up? Let me know out loud because I don't have the chat open. So if you need more time, let me know. Shout it out. If you're good, we'll move on. We got a couple experiments to do. All right. Now the ones that we can do live, we'll get results today. Dissolved oxygen. Okay, dissolved oxygen. You're going to use the other glass little tube, dissolved oxygen. This one. Grab the DO. Okay. Something that I learned yesterday with my students. You're going to take your tablets. Now, do not, this is the first time we're actually working with tablets. The first two, we didn't work with tablets until right now. Okay, do not try to pop these out like if it was medication. Don't try to pop it out. No, what you're gonna do is you're gonna like break the tablet, and we don't want that. If you see in your kits, you have paper clips. Why did I throw in paper clips? This is why. You're gonna open up your paper clip. You're gonna open up your paper clip so you have like a pointy little edge right there. Okay. Now, your tablets. What you're gonna do, let me stop sharing for a second so you see. Okay. Hello, I'm super big now. Okay. There is the side with no writing and the side with writing. What you're gonna do is you're gonna like go like around the tablet to like open up the foil. Go on the side with the writing. That's like the softer side. Okay. So what you're gonna do, and this takes time. You're gonna kind of go and poke a hole around it, not in the center, but around like where the tablet is. So I'm gonna poke a hole, and then I'm gonna poke a hole, and then I'm gonna kind of go around it so I can break the seal, kind of going around it. So you see how I'm going around it? I poke a hole, and then this is like really hard to show you. Poke a hole and then go around. Poke a hole and then go around, and then I can lift the aluminum with the paper clip and expose my tablet. Let me do it on this side. So poke a hole and then you're kind of going around. Notice I'm on the side with the writing on it, right? So all the way around, and then I'm gonna try to lift the aluminum. There you go. So I've exposed the two tablets, okay? So that's how we gotta use a paper clip. Do not try to pop them out. They're not gonna pop, and what you're gonna do is you're gonna break your tablets, and then you compromise your experiment. So do not just pop them out. Stick around, and then they're exposed. That's what we want. So let's see. Let's now read the directions. Fill a small test tube to overflowing. Okay, so I didn't read step 2, I'm just reading step 1. You're gonna fill the small test tube with water. Okay, so you go in there, you might have to hold it over, fill it up. The glass, DO, pour the water in there. Try not to make a mess. If there's air bubbles, pop them so you get the water in there. Experiencing some air bubbles. All right, so that's step 1. Step 1, fill it up. What's step 2? Add the 2 dissolved oxygen test tabs to the tube. Okay, so you're gonna add the 2 tablets inside. sure you don't drop them. So into the tube. So 1 and 2, and they sink to the bottom. And then cap the tube. Be sure no air bubbles are in the sample. So make sure you have water all the way to the edge. Add more water if you need it, all the way to the edge, and then put your cap back on. Mix by inverting until the tablets have disintegrated, about 4 minutes. Okay, when you shake, make sure you hold it so you have one finger on the cap. Even though you twisted, you can't rely on it. So put your finger on the top. You're gonna mix for 4 minutes. That's gonna be, for me, it's gonna be 10:34. So I'm gonna stop shaking at 10:34. So we're gonna be shaking for 4 minutes because that's what the directions say.
Ms. O'Malley,
Yes.
when we put the tablets in, or the you know what I mean? Do we fill up the little bottle, right? Not just halfway?
No. What does step number 1 say?
Oh, okay. Yeah, I didn't see that. Sorry.
Your best friend is those Google Slides, because I might go too fast or you might not hear a detail, but literally go back to the Google Slides and read each step one at a time. Don't read the whole thing. I'm only on step number 4. That's all I read. I didn't read step number 5. I'm just on step number 4, which is inverting until the tablets have dissolved, about 4 minutes. So I'm just shaking this for 4 minutes. So it's new, guys. 4 minutes.
Miss Lameli,
Yes.
is it supposed to be like a pink a little, like the color?
Yeah. If you look at the screen on, you see how on the right side it says 0, 4, and 8 on the Google slide? You see how it's like white, kind of.
Yeah, I see it.
Yeah. So those are going to be your results. So depending on what color you get at the end is your results. So yes, it should be changing colors. So these tablets that we put in there, they're testing for dissolved oxygen. How much dissolved oxygen is in here? And we just shake, shake, shake. Shake, shake, shake. Got 2 more minutes of shaking. And people get paid to do this, you know, in real life. People literally get paid to do this. So you're shaking. Oh, mine have dissolved. Oh no, they haven't. I have one more. God, this water smells like sewage. You can still see little bits of tablet. 1 more minute of shaking. Almost. Keep shaking it. Okay, that's been 4 minutes, and yeah, I think mine has dissolved. So yours I'm pretty sure all of yours has changed colors, right? Now, step number 5 says wait 5 minutes. Okay, so I'm gonna put this in my little kit on dissolved oxygen, and I'm gonna go, hey Siri, set a timer for 5 minutes.
Your timer is set for 5 minutes.
Okay, so we're gonna wait 5 minutes to see really what the results of this are. For me right now, it looks like a 4, like in the chart. I'm gonna see if it gets darker to an 8. It might turn into an 8 after 5 minutes. So we don't have results immediately because it might turn into a 4, okay? Are we okay? So that was the dissolved oxygen. Now we just wait for 5. I told Siri to set my timer. Feel free to ask Siri set your timer for you. Questions? Okay, while we wait for the 5 minutes, we're gonna start the next one, just so we use up our time properly, okay? So we're not done with the dissolved oxygen, but we gotta wait 5 minutes. So we're gonna go into the nitrate one. Oops. So for the nitrate one, you're going to use the plastic tube. So find your plastic tube. Now the cap can easily break off, so don't try to break it off. Okay, it closes, doesn't go all the way the other way. Okay, so this is your testing tube. Take a moment and look at it. You should see numbers on the side, on one of the sides. So look at the numbers. Do you guys see the numbers that are on the plastic tube? Thank you. Okay, so those are going to be important numbers to look at. All right, oops. So we're gonna do the nitrate one. These are the different colors that might appear for nitrate. Depending on what it is, we'll discuss later. Okay, step number 1. Fill the test tube to 5 ml. So find the 5. You can also, find the 5. See, 1, 2, 3, 4, 5. So do you guys see the line above the 5? That's where you're filling it. The line above the 5. The line above. Okay, so go ahead and fill it to 5. Fill it to 5. The line above the number 5. Okay, so fill your test tube, the plastic tube, to the number 5. We're okay? Add 1 nitrate tab, test tab. Okay, so what you're gonna do, close this up. Close it right now. Nice and tight. Make sure you hear the click, click. That means it's closed, okay? Lay it down. Why? Because you gotta get your nitrate. So if you look at your test tray, you should, on the nitrate, you should see tablets 1 and tablet 2. Do you guys see tablet 1 and tablet 2? If I'm going too fast, please let me know. I don't mind slowing down. So you should see, so I have my test tube full of water here. I put a cap on it right here. Now you should see tablet 1 and tablet 2. 1 and 2. Do you guys see those? For nitrate, it has the word nitrate on it, or part of the word nitrate. Step 2 says add the number 1. But yes.
I only see one. I only see number 2 for me.
Okay, so go to your other ones. Do you see a number 1 on the other ones? Like maybe it went to a different little container.
Mine did that as well. It was on a different container.
Okay,
There's pH and then P. And
Okay.
then the other one.
the oxygen one.
Yeah.
Okay, so do you not see a number 1 anywhere?
No. I only see it number 2.
Oh no. Okay. Darn. Okay, then you're missing that one. Sorry. I thought we double checked them. Okay, it's okay. I mean, you won't get all the results, but at least you get to try it. We'll get to experiment. Oh, that's 5 minutes. Okay, never mind. Stop. Okay, we're gonna put this one on hold, the nitrate on hold. 5 minutes was up. Go back. Sorry, if this is too confusing, I won't do this again. Go back to your DO. Go back to your DO. DO. Dissolved oxygen. It's been 5 minutes. So looking at your results for the DO, is it a 0, a 4, or an 8? What do you guys, if you look at your results, is it a 4, a 0, or an 8 of the glass one that we just did, the little one? Okay, so what you do is you go to this one, and this one's dissolved oxygen. And you're going to highlight, is it a 0 for you? Highlight. Come on, we can do the highlight. Is it a 0? Is it a 4? Is it an 8? What are you gonna highlight? So for the dissolved oxygen, everyone right now can answer the dissolved oxygen for your sample. Is it 0, 4, and 8 or 8? So highlight one of those answers for yours. So dissolved oxygen, what is the results for dissolved oxygen? Are you a 0, a 4, or an 8? After 5 minutes, you can determine that. If you go back to the slide, at the bottom of the card, it tells you what these results mean. Aquatic animals need dissolved oxygen to live. Fish, plants, bacteria all require oxygen for respiration. Oxygen dissolves rapidly into water from the atmosphere until the water is saturated. Once dissolved in water, the oxygen diffuses very slowly and distributes depends on the movement of the water. Oxygen is also produced by aquatic plants, algae. All right, so depending on the type of species, you're gonna need dissolved oxygen. Okay, levels below 3 are stressful to most aquatic organisms. So dissolved oxygen levels that are 2 and 1. So if you have like a clear, not pink, but like more white, more translucent, that means you have very little oxygen in your water. That means aquatic life cannot live in the water sample that you took because there's not enough oxygen for them. Won't support fishes. So if yours is clear or white, that means that it won't support fish. Levels of 5 to 6 are usually required for growth and activity of aquatic organisms. So if you have like a 4, like so mine would support aquatic life. Okay. So did anybody get like a white or clear?
I did.
You did?
Yeah.
Oh, that's so cool. Okay. Oh, which reminds me, take a picture. Take a picture of the results. Because
Holding my
that's
stuff and taking pictures for the extra credit assignment.
yes, but also, you're gonna take a picture of your results, like of the tube. You're gonna take a picture of the tube, and then you're gonna upload that to the worksheet. You're gonna upload that to the worksheet. Upload that to the worksheet. That makes sense? So I'm gonna take a picture of this and then I'm going to upload it on the second page right here. This is where I would upload the picture of my results so I can see your results. And I'm gonna be pretty cool to see like what Angel said, like, oh, mine was, is it white or is it clear, Angel?
It's like white. I can still see like the other side a little bit, but it's mostly white.
Okay, so yours turned white. It's not clear, it's white. Okay, I didn't know that.
Basically,
Cool.
like it has fog in the water, like that.
Wow, that's super cool. That means that there's not enough oxygen for living creatures to survive in the water. So like fish, the plants, the algae stuff, they need water. Your water won't support it. My water will. My water, we can support the algae, we can support fish and things like that. So we're all gonna have different results because all our waters are so different. But it's fascinating to figure out like, okay, what's up with yours? All right, cool. So you can go and highlight on your worksheet, keep track of your results. Super cool. All right, we're ready to move on to nitrate again. So nitrate, we already filled it up, right? You already have it filled up. Now, sorry, Billy, you don't have tablet number 1. I apologize. Don't know what happened. Okay, so step number 2 says add 1 nitrate tablet. So again, you're gonna use your paper clip, poke a hole, go all the way around-ish, or halfway, or 3/4, and then lift number 1. We're just doing number 1. We're going to lift it and expose the tablet. Okay, again, you don't want to try to pop it out. The aluminum foil is not thin enough to be popped out. You have to use your paper clip, carve around it. You have your tablet. This is tablet number 1. Tablet number 1. So it says, add 1 tablet, cap the tube, and mix until the tablet has disintegrated. Okay, so carefully open your container, drop in just 1 tablet, the number 1. Okay, close it, cap it, and mix. Now you want to make sure it's capped properly. You hear the snap. Now put your thumb or your finger, whatever, on top where the cap is, and now just mix it. Don't hold it like this, because if you don't cap it right, you're gonna have really ugly water all over your house. So cap, hold, shake until it dissolves. Okay, so we're shaking it. Remember, your tube should have 5 milliliters of water. Shake, shake, shake until it's dissolved. It's already dissolved for me. So now I'm going to read step number 4. Add 1 nitrate number 2 tablet. Okay, so now I'm going to again make sure it's closed, leave it on the table, pop, and then go around. Open it up, aluminum foil. Okay, number 2. So Billy, you can just try it with number 2, see what kind of results you get with number 2. Be curious to see. Okay, put the tab in there. Step number 5, cap the tube and mix for 2 minutes. Okay. Hey Siri, set a timer for 2 minutes. So cap and mix for 2 minutes. 2 minutes. So I put number 2 in there, and now I'm shaking it, making sure I keep a finger on top so I don't have water all over the place. That would be a no bueno.
How long are we shaking it, Miss Lomeli?
2 minutes.
Okay.
Reminder, if you have questions, just say it out loud. If you want to slow down, say it out loud. I'm not checking the chat because I'm only on one screen today. So Just let me know if you need anything. Shake, shake, shake. I'm glad we can still do this, even though we're not on campus. You guys are still able to test your stuff. All right, a couple more seconds. I think mine's dissolved, which is good. Ten seconds for me. I'm shaking. Okay, now step number 6 says wait 5 minutes, remove the, wait 5 minutes. Okay, hey Siri. Hey Siri, set a timer for 5 minutes. Okay, so this just gets to hang out for 5 minutes. You can put it in your tray. So on the nitrate to kind of wait. We can't do anything right now. We have to just wait. Make sure you answer the question for dissolved oxygen. Make sure you've taken a picture of your dissolved oxygen. So yeah, we just have to hang out because we're gonna need this tube again, but we gotta wash it out and we gotta wait for it for a second. So we're just hanging out for 5 minutes. What's new? So the results that we get, like for example, Angel, he got a result that's very different than my result. So he realized, well, once we do a little bit more research, is that there's not enough oxygen for fish. So next week, when we start working on our final presentations, that's something that if Angel chooses to do so, he can focus on because it's a problem that is occurring in his neighborhood. Like something is cutting off all the oxygen in that water, and we got to figure out what is it and how can we fix it. So the results that you get today in your experiments, whatever you test positive for, that's what you're going to use to write your report on. Like, okay, I realized that the water near my house in my storm drain has high levels of or low levels of, which means, you know, fish can't survive or there's a lot of pollutants. Here's a way to fix it, right? That's what we're doing these experiments. Okay, there's this problem. Okay, how can we fix it, right? Are you going to be fixing it by educating the population, like your neighbors, like, hey, we're throwing away too much, you know, food in the drains, so that's depriving our oxygen? Or is it something else, like the nitrates? Nitrates would be like your fertilizer. If your results show high levels of fertilizer, that means your neighbors or yourself or whatnot are using too much fertilizer. Okay, so that means we got to educate the people. So that's what we're going to be working on next week. With your results, you're going to determine, okay, what's the real problem? It's not just the trash that we see. It's not the leaves that we see. There's another layer that we didn't see until we tested. So you can check your nitrate. Mine right now is maybe like a 5, a level 5, like you can see on the screen right here, 0, 5, 20, 40. I'm curious to see if anyone's in a pop 40. Let me know if like your results show a 40. That's gonna be super interesting.
Lameli,
Yes.
for my nitrate, it's also foggy as well.
Foggy, okay. We'll see what happens in a couple minutes.
Okay.
We still have to wait 2 minutes. Things can dramatically change. If you're curious, we can start reading actually. This part. Nitrate is a nutrient that acts as a fertilizer for aquatic plants. When nutrients levels are high, excessive plant and algae growth creates water quality problems. Nitrogen enters the water from human and animal waste. Oh, so another poop. decomposing organic matter like food, and lawn and crop fertilizer runoff. So I just assume nitrate with fertilizer, but it also is in animal poop and organic matter, which organic matter is like food or anything organic. Food, like organic food, plants, those are all organic stuff. Unpolluted waters usually have a nitrate level below 4. So a below 4, so a clear or foggy, like no color, is actually a good thing. Levels of 40 are considered unsafe drinking water. So above 40, so if you get a super like red, pink color, not drinkable water. I mean, granted, you can't drink this whatsoever, but it has high levels of nitrates, nitrogens, animal waste, organic matter, fertilizer. Eee. So less than 4 is actually a good thing. More than 40 is a really bad thing. About 1 more minute and then we can see results. Let me see my results from yesterday. Okay, that was 5 minutes. You might be like a couple seconds after me. Mine's less pink than the one from the classroom yesterday. The one we did in the classroom was a lot more pink. So the school water had a lot more fertilizer than the water that I tested. So mine's like this. I would call it maybe like a 5. If you look at this, so let me go up. If you look at 0, 5, 20, and 40, I would say mine, for example, would be a 5. Take a picture. Take a picture. Take a picture of your results. Take a picture. Try to take a picture like where there's like a white background. Like if you have a plate and you see, if I put it on a white background, you guys can see the difference in color, right? If I put it on like my black shirt, it might not give you the same results. But if you look at a white background, you'll see it. Okay. Yes.
Mine's like the same as yours. When I put it on something white, it's like the same thing as yours.
Like a little yellow.
Yeah, yellow.
Yeah, I would like mine, for example, is like a 5. So yeah, I guess when we start looking at results, it would be a good idea to find something white. Even like, look, this is a styrofoam plate. Like I'm just kind of looking at my results. My first 2 results. Here are my first 2 results. You can see, right? The first one is a lot more pink. This one's a lot more yellow, but definitely not 20 or 40. All right. So take a picture and then you can go right now. Do it right now because you're going to dump this. You're going to clean out this tube. So go to this one, go to your results. And what we just did right now was the nitrate one. So this one. So are you a 0? Are you lighter than a 5? Or are you 20 or 40? So, again, going back to this, are like a 0? Are you 0 to 5? Are you a 5, or are you 20 to 40? So those are your options. Interesting. So Angel, like you have to make a decision. Are you like a 0 to 5 or are you a solid 5 based on these colors on your screen? And maybe you can hold your 2 to the screen and see like, okay, what colors am I lighter than the 5 or am I a solid 5 or am I darker? Anybody get a darker like pink? Anybody out there get like a 20 or a 40?
I have a question.
Yes.
For me, I think I have like a solid 5 cloudy because like when I see through it, I can't see anything, just cloudy. That's it. I can't see the other side.
Right. Yeah.
So I think I'm gonna put a solid 5.
Okay. So highlight that right now. Everybody highlight your results because we're going to dump these out. So your results, they're not going to stay there. Kind of like the DO, the dissolved oxygen, we're just leaving it there. It's hanging out. But this one, I need to reuse the tube. So make sure you do your results, highlight. You can insert the picture later, right? All right, we're good. Ready to move on? Okay, so we're gonna move on to phosphate. And with the phosphate, you're going to need 1 plastic tube wash after the experiment. Okay, so the phosphate, whoops. Phosphate, you're gonna fill the test tube to 5. Okay, so take this, the nitrate one, go to the sink, pour it out. Don't pour it over your dishes or go to the bathroom, pour it there. Don't pour it over your toothbrush. If you're gonna go to the kitchen sink, pour it into the drain. Don't pour it like that and then it lands on your dishes. So pour it directly into the drain and then wash it out with a little bit of water, no soap, just kind of rinse it, have some water running through it, just rinse it a couple of times and then it's ready to go. So let's do that right now. Either the bathroom sink or the kitchen sink. Dump the water, turn your water on, flush it out. Fill it with clean water, dump it out, shake it out.
We're doing this for the nitrate, right?
Yeah, the nitrate.
Okay.
Nitrate. So I just kind of rinsed it. And I like rinsed it out with water, no soap, just kind of a couple of times, filled it up with water, dumped it, fill it up with water, dumped it. All right. Grab paper towel, just kind of wipe it down a little. All right, we're ready. Oh, this one gives you a pretty cool color. All right. So step 1 says fill the test tube to 5. Okay, so it's cleaned out, it's washed out. Don't just dump it and come back. You gotta wash it out because we gotta get all that nitrate out of there.
Do we dry it?
No, it's fine. I just dried the outside. Don't worry about the inside. So fill it up to 5. Again, the 5 would be the line on top of the 5. People, like I said, people do this for a living. Like they literally what you're doing, someone does it for a living. They go out there, they collect samples, and then they test them, and then they give the results to the city. So I filled my clean test tube, plastic test tube with 5. I'm gonna close it. And then I'm gonna read number 2. Add 1 phosphate. Wait, is it phosphate? No, phosphorus tablet. Phosphorus tablet, which is the phosphate. It has a P on it. So grab the one that has the P for phosphate, not pH, but P. Okay, so here's my tablet. It has P, just the P on it. I'm gonna grab my paper clip. I'm gonna poke a hole.
Ms. Lamelli,
Yes.
I don't have one that says P. It says the whole word on the back.
Oh, does it say phosphate? Phosphates?
Yeah.
Oh, perfect.
Okay, thank you.
Just as long as it's not the pH one. Do you have a pH one on the pH? Separate?
Okay, so there's 2. One of them, it says PHOSPH, and then the other one, it just says PHO.
PHO. I think PO. Mine says pH something, and then the numbers are 542.
Yeah, 5422, and then I could slightly see half of an A.
Okay, that's the one we're using right now.
Okay.
Thank you for double checking. Anyways, if you don't get the color blue and you get the color purple, we knew it was the other test. Okay, so pop it open. Okay.
Actually, they both say the same thing. Phosphate, and they both have the same code.
Your other one should be 645. Oh, they do, huh?
I think there's two codes. Oh yeah, and then there's this other one that says 645. Yeah.
Okay, my pH says 645. All right. Anyways, I think Angel, the results will be totally different. So it'll be fine. The colors will be different. Okay. So step number 2, add 1 tablet, cap, and mix. Okay, so we're gonna open it, put the tablet in, close it, make sure it clicks, and then mix until dissolved. So this one doesn't give me a time right now. It just says dissolve it. So I'm just gonna shake it. And then once it's dissolved, we're gonna wait 5 minutes. Shake, 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 shake. All right, time, people. Do it.
We're shaking it to dissolve, right?
Yep. Shake until dissolved. Okay. We are going, okay, I'm good, I think. Okay, so it's 5 minutes. Siri, set timer for 5 minutes. All right, and then we wait. Just in case, so we'll have time to finish this one. I don't think we'll have time to finish the pH one. So what you're gonna do, so you have this, it's kind of waiting, it's kind of already turning blue for me. For the pH one, same thing, you're gonna, once you've recorded your results for the phosphate, you wash out your tube and you're gonna fill it up to the 10, which is like basically the whole tube. So you're gonna fill up later, fill up with water all the way to the top. You're gonna put the pH tablet in there, cap it, mix it, and it should give you immediate results. So this one is just the next one that you're probably gonna have to do on your own is immediate results. And then depending on the color, there's a bunch of colors for this one, is your results. So it's pretty cool. All right, going back to phosphate. So it acts like a fertilizer. When nutrients levels are high, excessive plant and algae growth creates water quality problems. Phosphorus occurs in natural waters in the form of phosphates. Over half of the phosphates in lakes, streams, and rivers comes from detergents. Oh, detergents, like the ones we use at the house. Phosphates level higher than 0.03 ppm contribute to increased plant growth. So when we use detergent on our clothes, we're adding phosphates to the environment. So if our levels are high, so the darker the blue, the higher the level of phosphates, which leads to excessive plant and algae growth, which creates problems. So the darker the blue that you get, bad, because it will then create excessive plant and algae growth. If you've ever had like a fish, you get like too much algae in it and it kind of smells too. Algae doesn't smell good. So the darker, bad. Here we actually want it to be clear. Darker is bad, bad. I'm sure your tube is turning blue, right? Right? Mine's turning blue. Look. See how blue mine is? And it will get darker. You have 2 more minutes to wait to see what it ends up looking like. Okay. So as we wait, just to recap, the glass tube, the big glass tube, that's on Sunday. You're gonna check your results on Sunday, the big glass tube.
Out of the sun.
What happened?
I said keep the tube out of the sun.
Yes. Anywhere like in a room, that's fine. Like just don't put it like near a window. The aluminum one, we're gonna do together Tuesday, right, in class. We'll uncover it on Tuesday, and then we'll add your tablets on Tuesday. So that's gonna be pretty exciting to do Tuesday together. So the aluminum one stays out of the refrigerator, like right next to the other one if you want. And we're just gonna leave it there. And you have those 2 tablets that you haven't used, okay? The DO, you can wash that out after class, okay? Make sure that you fill out your data sheet, okay? Take pictures. Ooh, mine's getting really blue. I want to say mine's right now, and I haven't even checked the time. I have less than a minute. Mine's a 4. If I hold it to the screen, mine's a 4, guys.
Mine too. It was like a light blue, and like I could see still clear it, but now it's just turned into a 4.
Yeah, which means plants can grow and algae can grow, but too much. So then it pollutes the water. Too much plants and algae. So it's not good, too much. Super cool. So, yes,
Are we gonna need to do pH ourselves?
yeah. The good thing about pH one is that you just shake it. You just shake it and you get your results pretty quickly. You don't have to wait. Okay, so right now that's the 5 minutes. So check the blueness, take a picture of it, go back to this sheet, and now it's the phosphate, it's the bottom one. Are you a 0, a 1, a 2, or a 4? For phosphate, it's the one at the bottom, not pH. We haven't done that one yet. And then of course, you can do this later, insert your pictures later. All right. So now that you've done the blue test, I don't think I took a picture of the other one. I mean, we have 4 minutes. We can do it. Go wash this out again. Wash out the blue. Once your 5 minutes, if you're like a little bit behind me, you still have to wait. If you're with me, we can wash it out. If you guys have like a real fish tank, like those really like, heavy, like glass fish tanks with fish in it, like sometimes you have to do tests on the water to make sure that the pH levels are okay and all the other levels are okay. Maybe you didn't realize that like your family member is actually doing that. You can ask them like, hey, do we test the water in the fish tank? Now, if you have like a goldfish fish tank that like doesn't have a filter, that's something different. Okay, so I cleaned it out, I washed it out, kind of just patted it dry. The last one is phosphate. Or no, last one's pH. Gonna use the test tube. Fill the test tube to 10. So that's like kind of all the way up. So do that. All the way to 10. All the way past. Remember, the line is on top of the 10. Almost all the way to the very, very top. Okay, cap it. Oh, cap it. Be careful. Cap it. Now you're going to open up the pH one. So again, you should be pros at opening these up now. Pop. And then around.
Ms. Lamelli,
Yes.
for my pH, it has the word range in the back. Is that for you?
I don't know. All I see is the code 645.
Yeah, code 645.
Yeah. Oh, it's kind of like a green tablet. Looks green when I opened it.
Okay, let me see.
All right, open it up. Open up your tube. I'm gonna pop the green in there. Green little tablet. Cap the tube and mix until the tablet has dissolved. So I'm gonna mix it. Oh, it turned green immediately. Oh, which might be a good thing. I don't know. pH test is one of the most common analysis in water testing. pH is a measurement of the activity of hydrogen ions in water sample. The pH scale, hold on.
I shaked mine for like 10 seconds and it already dissolved.
Right? Yeah, mine did too. Hold on. Okay. So mine's like right now it's a 7. So I was trying to read you the scale range of 0 to 14. Water samples with a pH below 7 are considered acidic. Those above 7 are basic, with 7 considered neutral. Okay, so below a 7, so if you get like yellow, pinks, it's very acidic. Purple is considered, purple is basic. So 11s and 12s, 11s and 10s are basic. If you get the yellows or the pinks, that means the water is acidic. So 7 is actually neutral. It's not too acidic, it's basic. 6.5 to 8.2 is optimal for most organisms. Seawater usually has a pH value of 8. Okay, cool. So yeah, I can say mine's a 7. So our acidic level is good, basic, neutral. All right, take a picture of that. Now, what you're gonna do is you're gonna check the big tube on Sunday. You can wash out your syringe. So your syringe, you can actually pop this out completely. Look, the syringe goes into, wash them out. No with soap, just water. Wash out the phosphate, wash out the dissolved oxygen, and just kind of leave them open to dry, right? You can just leave them on your tray, drying overnight. Because I do need these back because I'm going to use these next year with my, next set of students. So yes, what's up?
It's 11:17.
Yes.
Oh, okay.
I'm trying to finish up a lab.
Okay.
Yeah. So clean out this, clean out that, and then Tuesday when you take your yearbook picture, you'll turn that back in. Okay. So finish up your labs, clean up, make sure you clean up around you. It is dirty water. Throw away your gloves. You can throw away your water. Man, this water smells. Give it a good wipe. Clorox wipe would be good. If you've been touching your iPad and the water, clean your screen as well, or your keyboard. All right, guys. Any questions, please feel free to email me. If not, I will see you on Tuesday where we get to unwrap the aluminum foil. All right, guys. Any questions?
For DO, could we wash that already or do we leave it like?
Yeah, no. DO, you can wash it along with your pH.
Okay, so basically the only thing we're waiting for Tuesday or Sunday is the BOD and the big glass bottle, the one you're wearing.
The big glass bottle Sunday, but the aluminum is Tuesday. Aluminum is Tuesday.
Okay.
All right, you guys have a break. You still have a 10-minute break, so you're still good. Yay, science.